So good morning everyone. Today's our topic is about toxicity studies. So why these toxicity studies are important? First let us see. So what are toxicity studies? So when we have introduced any new molecule, so it has to pass through different stages. See, for example, if any molecule is discovered, so it will take around 12 to 15 years to release into the market. So before releasing into the market, it will go to different phases like preclinical trials as well as clinical trials. What are these preclinical trials? They are nothing but we will test that molecule on the animals before being tested in the humans. So those will come under the preclinical studies. So these preclinical studies are nothing but whatever the screening tests we will perform for the activity of the drug, as well as what we will do the ex vivo experiments, as well as the isolation studies, and all these comes under these preclinical studies. So preclinical studies are nothing but we will test the molecule on the animals, whereas clinical studies is nothing but after passing the preclinical studies. So when we test the molecule on different animals like rabbit, guinea pig, or and higher animals like monkey, so after that the molecule pass into the clinical trials. That means we will test that molecule on the humans. So these toxicity studies, why they are important means, see, if we have use directly the molecule, if you have released that molecule without testing, so that may causes various toxic effects on different organs. So in order to avoid those toxicities, so we are going to perform these toxicity studies, and these toxicity studies will comes under the preclinical trials. Not only that, during the clinical trials also we will perform concurrently. For example, chronic toxicity studies are there. So in that we will mainly measure the carcinogenicity of that molecule as well as genotoxicity of that molecule. So along with the clinical trials like phase 3 clinical trials, we can also perform these toxicity studies. So coming to the introduction, so these toxicity studies will reveal the drug effects. So whatever the drug or whatever the molecule will causes the changes in the functions, or if it damages the tissue, so those changes will be detected by these toxicity studies. So the first point is that, so these toxicity studies will reveal the physiological as well as histopathological changes which are induced by the drug or the molecule. And next one is coming to these toxicity studies, they are mainly like three types: acute toxicity, subacute, as well as chronic toxicity studies. So these acute toxicity studies, they are the single dose studies, and they will determine the qualitative and quantitative assessment of the toxic reactions. And coming to the chronic and subchronic toxicity studies, they are the repeated dose toxicity studies, and they will characterize the toxicological profile of that molecule by following the repeated administration. So in acute toxicity studies, it is a single dose administration. Okay, so it will reveal the qualitative and quantitative assessment of the toxic reactions of that molecule, whereas chronic and subchronic toxicity studies, they will give the whole toxicological profile of that molecule. So according to the European Union, they have given the minimum duration or the time interval for these toxicity studies with the base of this duration of the clinical trials. For example, if the clinical trial we are going to use a single dose, so in rodents the minimum duration of these toxicity studies should be like 2 weeks. In non-rodent species, means other than the rodent species like rabbits, so we can test for 2 weeks. So if the duration of the clinical trials is up to 2 weeks, then the minimum duration of the repeated dose toxicity studies is 2 weeks, as well as in non-rodents also 2 weeks. Coming to this, if the duration of this clinical trial is 1 month, the minimum duration of these toxicity studies should be done like 1 month in both rodent and non-rodent species. And if we are going to perform the clinical trials up to 3 months, so then the minimum duration of these repeated dose toxicity should be 3 months both in rodent as well as non-rodent species. And if the duration of these clinical trials is more than 6 months, then the minimum duration of these repeated toxicity studies should be 6 months in rodent species and for chronic period in non-rodent species. So this is for if you observe, this is the single and repeated dose toxicity requirements in order to support the phase 1 clinical trials and in patients phase 2 according to the European Union. And here it is the repeated dose toxicity requirements to support the phase 3 trials. Okay, before we have seen phase 1 and phase 2 trials, the requirements, and now the repeated dose toxicity requirements in order to support the phase 3. Okay, so if the duration of the clinical trial is up to 2 weeks, then the minimum duration of these toxicity studies should be done for 1 month in both rodent as well as non-rodent species. And if the duration of the clinical trials is like 1 month, the minimum duration for conducting these toxicity studies should be like 3 months in both rodent and non-rodent species. And if the duration of the clinical trial is 3 months, like up to 3 months, then the 6 months toxicity studies has to be done in both rodent and non-rodent species. And if the duration is more than 6 months, so then the minimum duration of these toxicity studies should to be performed is like 6 months in both rodent, and in non-rodent it varies like chronic period of time. So chemicals, they are first tested in order to estimate this LD50, that is lethal dose 50. That is nothing but the dose which is required to kill 50% of that organisms. So we will select 2 animal species for testing, and we can use 2 routes of administration. In that, one route is intended for the purpose. Means, for example, if the drug is if the molecule we discovered is going to give in the IV route, so that route should be included in this. Along with that route, another route should also be followed. So 2 route of administration should be followed and 2 animal species are used. So as I said, one of this is the expected route of exposure of the human beings of the chemical being tested. That means the chemical which we are going to test and that will be taken by some route known like oral or IV. So that route, along with that route, that chemical should be given in the other route also. So the number of animals which die in the 14 days after the administration of this single dose has to be noted. So the animals which also examined for the signs of intoxication as well as weakness and mortality and morbidity rate also, as well as they will also observe the behavioral modification in these 14 days. So this is the acute toxicity studies. So in acute toxicity studies, so we will administer the drug in single dose. Only we will give in single dose, and after that we will observe the behavioral effects as well as the signs of the morbidity or mortality as well as the behavioral modifications in these 14 days we have to note. And after the acute toxicity studies, next the chemical has to be tested for 90 days. So this is the subacute toxicity studies. In acute toxicity studies we have tested only for 14 days, and here in subacute toxicity studies we are going to test for 90 days. And this is also performed in 2 species of animals, and also we can use the 2 different routes, and at least 3 doses has to be employed. As this route is the repeated dose toxicity studies, means as we can administer at least, we should administer at least 3 times. Means we should administer at least 3 doses. So in this way we will monitor a variety of parameters, like at the end of these studies we will also examine the organ and tissue also. So these organ and tissue will be examined by the pathologist. So in acute toxicity studies we will perform for 14 days, and after the after performing for 14 days we have to note the behavioral changes also. And not only that, we will also observe the signs of lethargy or intoxication. And here next to the acute toxicity studies, this is the subacute toxicity studies. We will perform for 90 days, and in that 90 days we should note all the changes. And at the end of this period, means at the end of these subacute toxicity studies, we have to examine the organs also. So the organs and tissues will be examined by the pathologist. And after the subacute toxicity studies, there is the chronic toxicity studies. So these chronic toxicity studies are the long-term studies. So the drugs can be exposed, means for the long period of time. So if the drug normally will be exposed for short period of time, for example in case of antimicrobial agents, and some drugs, some molecules can also be studied for more than 6 months. So a study of 6 months is sufficient in case of long exposure of the drug. For example, if the molecule which was discovered is intended only for the short term courses like antibiotics, then the short time interval is sufficient. But if the drug is going to administer for the long periods, like long durations like anti-cancer drugs or such kind of drugs, we have to do the studies at least for 6 months. So study of 6 months might be sufficient if the drug in the humans can be used for the longer periods. And a study of the chronic use can like 2 years might be also required. So normally the chronic toxicity study period is like 6 months, but if the drug is intended for the long term use or the chronic use, we can also study for 2 years. And coming to this chronic toxicity studies, we will also determine the carcinogenicity as well as mutagenicity properties as well as genotoxicity of the drug. What is this carcinogenicity? That means the ability of the molecule or the ability of the drug to cause the cancer like conditions is known as carcinogenicity. And mutagenicity means the ability of the drug to cause the mutations, that is known as mutagenicity. And next one is this genotoxicity, means some molecules can also affect the functioning of the genes, so that studies is known as genotoxicity. So coming to the chronic toxicity studies, we will determine, means we can determine the molecule for the mutagenicity, carcinogenicity as well as genotoxicity also. So in this we will also perform the Ames test. So mainly the Ames test is to determine the carcinogenicity or mutagenicity of the drug. So mainly for mutagenicity, the Ames test is preferred. So in these teratogenicity studies are also be performed by administering the molecule to the pregnant rat or pregnant rabbit during the period of the organogenesis, means during the development of the organs in the fetus in the rabbits or rats. So the molecule will be administered to the pregnant rats or rabbits. And the teratogenicity means the ability of the drug to cause the disabilities in the fetus is known as teratogenicity. So that can also be studied by these chronic toxicity studies. So as I said, for studying the mutagenicity, the test we will perform is like Ames test, and this test was discovered in order to study the reverse mutation, and this test was discovered by the Ames and the colleagues in the year 1975. So in this test they used the strain of Salmonella typhimurium. So it is also one of the causative agent which causes the typhoid fever. Salmonella typhi, same like that, another organism also there which causes the typhoid fever, that is Salmonella typhimurium. So this Salmonella typhimurium, it has a mutant gene, okay, for the enzyme known as phosphoribosyladenosine triphosphate enzyme. So this enzyme is required for the histidine synthesis, and this bacteria known as Salmonella typhimurium won't grow in the histidine deficient medium, means it requires histidine for the growth, but it also contains a mutant gene like phosphoribosyl adenosine triphosphate synthase, and in the absence of this enzyme, so there will be no synthesis of histidine, because this enzyme is responsible for the synthesis of histidine, and in the absence of this enzyme there will be no synthesis of the histidine and there will be no growth of that organism. So if we are going to use the chemical in the media which we are going to grow this Salmonella typhimurium, if that molecule is going to cause the mutations in the genes, so that can produce the gene which encodes for this enzyme, and if this enzyme is produced, it can synthesize the histidine. So again I am saying, normally this enzyme like phosphoribosyl adenosine triphosphate synthase, so this enzyme is required for the histidine synthesis. So in the absence of the histidine, so this organism won't grow, means if we have supplied this means we have inoculated this organism in the media which is deficient in histidine, then this organism may not be grown. So if we add a chemical that causes the mutations and that causes the changes in the genes, and if that chemical causes the mutations in the gene which encode for this enzyme, then if this enzyme is encoded, if this enzyme is produced by the gene, so then this enzyme can produce the histidine. And most of the chemicals are not mutagenic as well as not carcinogenic unless they are activated by the enzymes which are present in the endoplasm reticulum as well as or in the presence of the rat hepatic microsomes. So whenever we add these types of enzymes which are present in endoplasm reticulum or microsomes, that is rat liver microsomes in that media, then only they get metabolized and they can lead to carcinogenic or mutagenicity effects. So this Ames test is rapid and as well as sensitive, and it is mostly used for detection of the genotoxic carcinogens like benzopyrin and aflatoxin. What is this genotoxic carcinogens? Means the cancer causing agents which can also which is also toxic, which can also causes toxicity to the genes, are known as genotoxic carcinogens. So benzopyrin, example is benzopyrin as well as aflatoxin. So in case of detection of these chemicals, this test can be used, and it does not detect the non-genotoxic carcinogens. Only this will give the prediction for the genotoxic carcinogens, and it won't give, we cannot determine the non-genotoxic carcinogens. So coming to the summary, so acute toxicity studies, they are the short-term studies which are intended only for 14 days, and subacute toxicity studies, they are intended for 90 days. And in the subacute toxicity studies, at the end of that study, the organs and the tissues will be examined by the pathologist. Coming to the chronic toxicity studies, we can determine the carcinogenicity as well as mutagenicity, and not only that, teratogenicity as well as mostly carcinogenicity and teratogenicity. Not only that, mutagenicity can also be determined. Not only that, we can also determine the genotoxicity. So all these effects can be determined by these chronic toxicity studies, and the test which we can use, which we can predict the mutagenicity, is known as Ames test, and that is developed by Ames and the colleague in the year like 1975. So they use a strain known as Salmonella typhimurium, and that organism won't grow in the histidine deficient media, and if the chemical we are going to test, if that is added in that media, that chemical can causes the changes in the genes, and that gene if they causes the changes in the gene, and if that gene can encode the enzyme that is responsible for the synthesis of the histidine, then there will be growth of the Salmonella typhimurium. So this is about the toxicity studies.